Hello everyone, it's day 70 and today I have May 12 Indian Express, or not Indian Express, the Hindu newspaper, and I am on the science page because this is Sunday's edition. So there are a few important article over the Hindu page, in the Hindu newspaper over the science page. So let us see quickly one by one two important articles over this page. One is regarding the will cattle be the next lab for flu host switching. So let us see. So, cattle have so far not been associated with being infected on large scale, and as a result, cattle have not been well studied as domestic host for influenza A virus species. In contrast to the notion that cattle are considered to be the almost resistant to infections with influenza A virus H5N1 virus, which was first detected in dairy cattle in Texas in late March, has rapidly spread to 37 herds in 9 states in the US as of May 7. On April 24, the US FDA said that in a nationally representative commercial milk sampling study of pasteurized milk, about 1 in 5 of the retail samples tested positive for bird flu viral fragments. A greater proportion of positive results were in milk from areas with infected herds. An NIH-funded study has found an absence of infectious virus in milk samples. The April 23 report of the FAO noted that the H5N1 virus was detected in high concentrations in milk from infected dairy cattle and at levels greater than that seen in respiratory samples. That the concentration was less in the respiratory samples of the infected cows compared with the milk samples strongly suggested that the pathogenesis of the H5N1 virus in the cattle differs from other mammals, says a study posted in a preprint. Preprint Preprints are yet to be peer reviewed. One reason for dairy cattle milk containing high concentrations of H5N1 virus fragment could be the propensity of the virus to infect the mammary glands of cows, as a previous study has found. On evaluating the expression of H5N1 receptors in the mammary gland, respiratory tract, and cerebrum of the cattle, the authors found both the humans and the duck receptors to be highly expressed in the mammary glands. In the mammary gland, the human receptors and the duck receptors were found to be widely distributed in the alveoli, but not in the ducts. Chicken type influenza receptors were common in the cow respiratory tract. The high concentration of H5N1 virus fragments the milk from H5N1 infected cows could be due to the local viral replication in the mammary glands of the cows. H5N1 has high affinity for the receptor, the author says. The study found that the chicken receptor was expressed on the surface of the respiratory epithelium in the upper respiratory tract and upper part of the lower respiratory tract, while human and duck receptors were either lacking or very limited in expression. However, in the lung alveolar cells, the researchers found all the receptors of humans, chicken, and ducks being abundantly expressed. The abundance of human and duck receptors in the mammary glands and the large presence of the human, chicken, and duck receptors in the lung alveolar cells of cows provides a perfect environment for the evolution of H5N1 viruses that can easily spread from animals to humans. The reasons why pigs are called the evolutionary lab for flu host switching is precisely due to the presence of both the human flu and the avian flu host cell receptors in their upper respiratory tract, says Dr. Sam Scarpino from Northeastern University in a tweet. The latest study has found that the cow mammary glands contain the same kind of mixed flu receptors seen in pigs. One of the key changes required for the avian flu to transmit effectively in humans involves flu's hemagglutinin HA host cells receptor preference, Dr. Scarpino said in another tweet. Currently, the cell surface receptor that influenza uses in birds is subtly different from the one in the human upper respiratory tract. But when pigs get infected with human and avian influenza virus at the same time, the viruses can potentially undergo reassortment, where in small segments of their genomes get swapped. The swapping might sometimes help the avian flu viruses to become better adapted to bind to human receptors and hence spread from birds to humans more easily. The H1N1 pandemic of 2009 was due to the reassortment of the virus in the pig populations. The co-expression of the both humans and avian receptors in the mammary glands indicate the susceptibility to the virus of both swine, human, and avian origin. The co-expression of both receptors can make bovines to behave like a mixing vessel for new influenza A virus with increased zoonotic potential, the authors write. With the mammary glands of cows harboring receptors for both human flu and avian flu, dairy cattle may have similar potential as pigs to serve as evolutionary intermediaries between avian and human flu. So this was about. So let us see another important article over the 30 years study links ultra processed food to risk of early death. So higher consumption of most ultra processed foods is linked to a slightly higher risk of death with the ready to eat meat, poultry, and seafood based products, sugary drinks, dairy based desserts, and highly processed breakfast foods, showing the strongest associations, finds a 30 years US observational study. The results were published in the journal the BMJ. The researchers say not all ultra processed food products should be universally restricted, but that their findings provide support for limiting consumption of certain types of ultra processed food for long term health. Mounting evidence linking ultra processed food to higher risk of obesity, heart disease, diabetes, and bowel cancer, but few long term studies have examined links to all causes and cause specific deaths, especially due to cancer. To address this knowledge gap, researchers tracked the long-term health of 74,563 female registered nurses from 11 states in the Nurses Health Study 1984 to 2018 and 39,501 male health professionals from all 50 US states in Health Professionals Follow Up Study 1986 to 2018 with no history of cancer, cardiovascular diseases, or diabetes at study enrollment. Every 2 years, participants provide information on their health and lifestyle habits, and every 4 years, they completed a detailed food questionnaire. Overall dietary quality was not assessed using the Alternative Healthy Eating Index 2010 AHEI score. During an average 34-year follow-up period, the researchers identified 48,193 deaths, including 13,557 deaths due to cancer, 11,416 deaths due to cardiovascular diseases, 3,926 deaths due to respiratory diseases, and 6,343 deaths due to neurodegenerative diseases. Compared with the participants in the lowest quarter of ultra processed food intake, average 3 serving per day, those in the highest quarter average 7 servings per day, had a 4% higher risk of total deaths and 9% risk of other deaths, including an 8% higher risk of neurodegenerative deaths. No associations were found for deaths due to cardiovascular diseases, cancer, or respiratory diseases. In absolute numbers, the rate of death from any cause among participants in the lowest and highest quarter of ultra processed food intake was 1472 and 1536 per 100,000 persons year, respectively. The associations between ultra processed food intake and death varied across specific food groups, with meat, poultry, and seafood based ready to eat products showing the strongest and most consistent associations, followed by sugar sweetened and artificially sweetened beverages, dairy based desserts, and ultra processed breakfast food. The association was less pronounced after overall dietary quality was taken into account, suggesting that dietary quality has a stronger influence on long term health than ultra processed food consumption, note the authors. So, this was about the science that was covered on the Hindu newspaper on every Sunday. So we'll meet very soon with the May 13 Indian Express newspaper. So till then,